Thanks so much for coming to listen to the talk. I'm going to be discussing a project I've been working on for a while. It's very much still a work in progress, so comments, suggestions are welcome. So this is about premenopausal breast cancer risk prediction, and I'm using data from a large consortium. So premenopausal breast cancer or breast cancer is a leading cause of cancer diagnosis among women who are under 50 years of age. And the rates for women under 50 years of age have actually been sort of steadily increasing over the years. This is from SEER data taken up through 2018 with about a 0.5% annual change. So the breast cancer risk factors differ by menopausal status. There has been evidence of this for a few different risk factors. On the left here, you can see weight as a risk factor. And if we look at the categories obese, average, and lean, we can see that when a woman is younger, the incidence of breast cancer is actually lower among the obese category compared to the lean category. After a certain age, after age 55, that does reverse. So this is one of the distinctions between pre and postmenopausal breast cancer risk factors. I also wanted to point out on the right here, we have how age at first birth influences the time until a woman feels a protective effect from that birth with regards to breast cancer. So an earlier age at first birth results in a quicker time into which a woman then has this protective risk from the birth. So this is another risk factor that can differentiate between pre and postmenopausal women based on the timing. Other risk factors that are less explored for premenopausal women include breastfeeding, recent weight change, and oral contraceptive use. And so we'll be exploring those as well. So I'm using the premenopausal breast cancer collaborative group data. It's a consortium of 19 cohorts with data collected for women up to age of menopause or age at 55. And the populations represented include European populations, Canada, the US, Japan, Australia. There's wide diversity there. They've had some recent publications. And I just want to point out this is the collaborative group, all of the studies that are included. And I'm highlighting here the number of subjects, cases, and the number of follow-up rounds. So you can see we have multiple rounds of data. So we are updating our data over time. And we do have over 21,000 total cases to work with in this consortium, which is huge for premenopausal breast cancer. So our goal here was to develop a 5-year risk prediction model for premenopausal breast cancer. And I'm using a Cox proportional hazards model for 5-year risk based on the Rosner-Colditz model that has been updated in 2017 for overall women. And this model is going to be stratified by cohort, and I'll use backward selection to choose variables into the model. So the first stage, based on data access, is to develop the model within 4 cohorts that I have access to, and divide this into 2/3 training and 1/3 test data in order to look at the performance of the models. In the second stage, I'll expand it to all of the consortia, all of the cohorts. The total cases we have in the 4 cohorts I'm working with now are 5,175, which is a pretty good number. So the main thing I'm going to talk about today is how we're handling missing data in this project. So the missingness for each variable of interest differs by each cohort. And there are some cohorts that are entirely missing a variable of interest that we want to include in our risk prediction or think is important to at least assess. So for example, the sister study has no information on BMI in young adulthood. If we use sort of a complete case approach, we entirely lose the sister study from our risk prediction model. And this really limits the information that we can gain from having such a large consortium. So we thought of a way to incorporate missing data specifically for weight in young adulthood and have applied this to our current protocol. So if a woman has entered a study between ages 18 and 24, then she's assigned weight at childhood or in young adulthood as the same as the first weight of entry, which makes sense. If she doesn't have that information and she doesn't have a weight in young adulthood recorded separately, but she does have shape in childhood from a pictogram, or has relative weight in childhood recorded from the cohort, then we've decided to fill in her BMI at age 18 based on the percentile of the birth cohort in which she is from. So if a woman at her most recent weight was in the 50th percentile of the recent weight in the cohort, then we would fill in her BMI at age 18 as the 50th percentile according to the birth cohort at which she resided. So if we then have a couple more options we cannot impute. We can fill in from the most recent weight available and sort of backfill, or we can fill in from the distribution of weights from age 18 to 24 by the birth cohort. So like I said, corresponding with percentile at most recent weight. So I've applied these measures and compared sort of how it changes our results. Comparison is given here. So I have no imputation in which the sister study is completely unrepresented, imputed by just shape in childhood if they have that information, and then finally imputed by the distribution based on recent weight. And we can see here that we gain a lot of information when we do the imputations, and the current weight, the BMI in young adulthood, and the 4-year weight change, these variables of interest that we want to include in our models, the means and standard deviations do not change that much. So I've had to make a couple other assumptions. So this is for variables that are missing within cohorts, but that aren't completely missing by design in a cohort. So for instance, age at menarche was filled in with the median age at menarche where it was missing alcohol consumption. I filled it in with median alcohol consumption if the person was a current drinker based on their cohort. And then oral contraceptive use is filled in by age of or end age of use, as well as questionnaire age. And breastfeeding duration is replaced with the lowest category, and it's treated as a categorical variable. I did compare the results to a complete case model using all original variables to see how these additional assumptions affected the results. So the model selection process, I started from the variables that are included in the Rosner-Colditz 2017 model: current age, age at menarche, parity, age at first birth, height, current BMI, BMI in young adulthood, alcohol intake, family history, and benign breast disease history. So I'm using backwards elimination in order to find which variables are significant here, and then adding in oral contraceptive use, recent weight change, and breastfeeding variables. Again, backwards elimination was used to determine which should stay in the model. And eventually we get to our final model. So now I'm going to explain the models that were produced. So this is the complete case version where I did no imputation on any variables. These are all original variables in the data set. And I had 2,023 cases in my training data set to work with. So the variables that get selected into the model include current age, age at menarche, parity or birth number, height, current BMI, BMI at age 18, family history of breast cancer, history of benign breast disease, and current oral contraceptive use. And we can see that this closely aligns with the variables that have been selected for postmenopausal risk models, but there are some differences that are interesting here, especially the addition of current OC use and the opposite direction that current BMI is associated with risk among premenopausal women versus postmenopausal women. So keeping this in mind as our complete case model, I'm going to show a comparison of the models based on imputation method. So on the left is our no imputation of weight. So this is based on the imputation method that we use for young adulthood weight. So with no imputation of weight, I have 2,568 cases in my training set. And all the ORs and the variables that get selected into the model based on the procedure are the same as those selected in the complete case, and the estimates are very close to what the complete case found. Again, this is completely ignoring the sister study contribution since they do not have this information. So after imputing based on shape and childhood, if they had that available, the same variables are selected into our model. And I think this is important because it demonstrates that perhaps the imputation by shape is not erroneously like showing something that we shouldn't be seeing. In the next column is imputation by the percentile of the birth cohort defined by recent weight percentile. And this we get the most cases, 2,920. And we do see a slight difference here in that oral contraceptive use was no longer significant in the model, and so it wasn't selected for the final model. At the bottom here, I have the concordance statistics for the training and the testing data set. They're about 0.67, 0.68 for all of the models, and there aren't stark differences here. So I also wanted to evaluate oral contraceptive use as a past, never, and current use variable. And when I tested this and replaced this variable for current oral contraceptive use, past use appeared to have no effect, and the other variables that were included in the models did not change effect estimates. So as you can see here, in both the models that had oral contraceptive use as a variable selected, the current use is associated with an increase in risk, whereas the past use is not associated with any risk. So next I stratified by current age. So this is the questionnaire age. And we are doing this by 5-year risk models. So this gets updated every 5 years for each individual. So if you're under 40 years of age, we had 591 cases in our training data set. And 40 to 45, we had 920. 45 plus was over 1,000. So I just want to point out some of the differences we see here by the current age. And this may be very interesting when we continue to assess these models. So for instance, current age appears to be much more influential when a woman is younger versus when she's above age 45. So it's still significant in any model. Here we have age at first birth, which you can see looks to be slightly protective in women who are younger than 40, but slightly harmful in women who are greater than 45. And it didn't really have an effect in women in between. So this is a variable that is interesting and may reflect the change over time that occurs from what I showed you at the beginning of the graph of age at first birth, influencing when the protective effect kicks in. So birth number did not appear to be important for younger women, though it did influence risk in older women and had a significant reduction in risk. Breastfeeding duration did not get selected until women were over 45. And in this case, it looked like any category of breastfeeding duration resulted in a slight decrease in risk. BMI is an interesting variable here. For less than 40 years and 40 to 45 years, current BMI was associated with a decrease in risk. At 45 plus, BMI was not selected in the model, but BMI from age 18 to 24 was, and it was associated with a decrease in risk. This may represent sort of the correlation between current BMI and BMI at age 18 with respect to the age groups here. The alcohol and drinks per week was associated with an increase in risk, but only for those who were above 45. So I think it's important to think about the differences here between the age groups. One more example I'll describe is family history of breast cancer, which was much higher for those less than 40 years of age with an OR estimate of 1.93. So the takeaways from this, we lose a significant amount of information when we exclude an entire cohort due to missing by design, and we need to figure out a way to resolve that issue. So within cohort imputation assumptions that I used when a cohort had less than 100% missing for a variable, altered point estimates slightly when I checked them compared to the original variables, but did not alter selection of variables into the models. The imputation strategy for variables missing by design may impact the final selection based on the participants that end up being included in the cohort. So you did see that using the imputation of birth cohort weight distribution based on recent weight distribution, we didn't get OCUs in our final model, whereas a different imputation method just by shape and childhood, we would see that in the model. So it's important to think about who's being included and excluded here. It's also important to note that the effect size didn't change drastically, but the significance value did change for the oral contraceptive use. Family history and benign breast disease history have a much stronger impact on premenopausal breast cancer risk than any other variables found here. We didn't see breastfeeding or for-year weight change influence risk except in the older age group. Current BMI is associated with a very slight reduction in risk, which contrasts with postmenopausal breast cancer models, which is something interesting to keep in mind. It wasn't just BMI in young adulthood, it was also current BMI that appeared to look protective. We did see the effect of predictor variables varying by age group. And I want to talk a little bit about next steps. So where is missingness concerning in the full consortium? So we talked about sort of the plan used to impute variables for BMI in young adulthood. However, there are several other variables that have a large amount of missingness within the cohorts. So alcohol and benign breast disease history are missing in 10 cohorts, and breastfeeding duration is missing in 6 cohorts. So these are variables that we'll also have to make decisions on for how to impute due to their missing by design nature. So there are some alternative methods for handling this missing data. We could impute based on a reference data set that has similar demographics and has the full set of variables available. For the US, we might use NHANES. For European populations, we might use Health Survey of England. There are a couple issues, and the reason why we didn't start out doing this is because the data set for NHANES, for instance, does not have full data on the variables of interest here, especially the interesting variables that sort of we wanted to add to our models. So BMI in young adulthood was not included, which precluded the use of for-year weight change. And also breastfeeding is not included. So if we wanted to use the NHANES data set, we would first have to impute these variables into the NHANES data set based on the data that we already have within our cohorts, which is kind of circular and might not be the best method. So the demographics of larger reference data sets might not match what the demographics of the cohorts are. We can remedy some of this with weighting, but not all of it. Um, and basically, we just have to keep making decisions on how to best use the full data that we have in the 19 cohorts in order to get an accurate risk prediction model for premenopausal breast cancer. So that is all I have for you. Thank you to Heather, my mentor. Bernie Rosner has helped a lot with this project. Rula Tamimi, who's now at Weill Cornell, a premenopausal breast cancer collaborative group, breast cancer working group at Channing, and grant support from the T32. And thank you for listening.
Hello, everyone. My name is Kai Wang. I'm a postdoc working with Dr. Miyang Song. Today, I will discuss a project, the gut microbiome modulates body weight response to physical activity. I will expand my talk in the following sections: background, research questions, the design, results discussion, and strengths and limitations. Background. We know the physical activity offers a cost-effective way for energy expenditure, so it's often prescribed for weight control. But physical activity does not always result in expected weight loss, and it shows large individual variability in the body weight response. According to this study, 8 weeks of exercise intervention, after the intervention, the participants shows a variability in their change in BMI and fat mass. And we may define this group people with a well response as responders in their body weight to physical activity, and the other group that didn't respond well as non-responders. There are several potential mechanisms proposed to explain the variability, but the two most often discussed modifier are the modified appetite after the physical activity and the perceived reinforcement value of food, which means the participants are less willing to intake food after the physical activity intervention. However, according to a study, they found after 7 weeks of physical activity intervention, there's variability in the fat mass change, but even after the controlling for the net exercise energy expenditure and the changes in energy intake, there remain a substantial variability in their fat mass change. This evidence points to that there should be other modifiers of the response not identified. The most straightforward mechanism for obesity is the imbalance between energy intake and energy expenditure, but there's also a large body of evidence demonstrating the gut microbiome also plays an important role in energy balance, including with their mechanisms, including the gut microbiome induced changes in energy harvesting, in metabolic pathways, in inflammatory response, in brain and in behaviors. All this points to that the gut microbiome may predispose the development of obesity. For the physical activity and the microbiome, there's also a number of studies performed with the main finding is the exercise may influence the microbial composition, function, and the microbial metabolism. However, how the changes in the microbiome may modify their exercise influence on the other diseases or disorders such as IBD, colorectal cancer, obesity remain unclear. So this is based on this background and the literature gap, we raised our research questions. First, we want to know whether the gut microbiome modulates body weight response to physical activity, and we hypothesize that the individuals with certain microbial features will have a better response in body weight than the other group. And we also want to know what specific microbe in the gut microbiome drive their modification. Because of their increased energy expenditure by physical activity, we hypothesize it is the microbes in their energy metabolism may drive this modification. Now, we also want to know what specific functions in their microbes drive their modification. and we hypothesize it's the pathways involving glucose and lipid metabolism. We designed our study in the Men's Lifestyle Validation Study, MLVS. It's a sub-study in the mother cohort or health professional follow-up study. In HPFS from the baseline of 1986, the information of physical activity and body weight was collected every 2 years, and in MLVS, 2 pairs of stool samples and 2 blood samples were collected 6 months apart, and accelerometer and double label water was also administered for to monitor the recent physical activity and for the calculation of fat mass percentage. We performed our core analysis in the discovery cohort or MLVS, and we additionally have an independent validation cohort mind-body study. So based on this design, we have the exposures of physical activity in met hours overall and by intensity, and we used the body weight change from age 21 to the time of stool collection as a primary outcome. We also use a BMI, fat mass percentage, plasma CRP, HbA1c as the time of stool collection, as well as the short term, it's 6 months with change between the first and second stool collection and the secondary outcomes. And we use the microbiome abundances of species as well as metacyclic pathways and EC enzymes as a potential modifier. We designed 6 associations to examine this study. They are the recent physical activity, which is represented by the physical activity data collected by accelerometer in relation to BMI, fat mass percentage, plasma CRP, and HbA1c as a time of stool collection. And the physical activity as the first stool collection in relation to the weight change between the 2 stool sample collections, as well as the long-term physical activity in relation to a long-term body weight change from age 21 to stool collection. And we use the average community average across all the follow-ups to represent the long-term physical activity. Our results, first we described the change in BMI from the age 21 to a time of stool collection. We observed a substantial increase in the BMI, and the BMI reached the peak around the age of 65. We also examined the correlation between the major variables. We found recent total physical activity and the long-term total physical activity are corrected at a low magnitude with a coefficient of 0.4, and the long-term and the recent physical activity are largely in negative correlation with the adiposity measures. We then examined the overall microbial structure using PICO analysis. We found short-term and long-term physical activity are not major driver of the overall microbial structure. We also examined the specific contribution for each variables and covariates to the overall microbial structure using Permanova analysis. We found the recent total physical activity, long-term total physical activity, BMI, and the probiotic use share a similar proportion to the variation in their structure, although we didn't observe significance for some. We then examined their per feature association of their major measures, and the most outside layer shows the association of recent total physical activity with the specific microbes. And then is a long-term physical activity, and then is BMI, then is fat mass percentage, and then 6 months with change, and then is long-term with change. Largely, we found the physical activity, the association of physical activity are at opposite direction with the association with the body weight measures. And we also found physical activity was associated with a specific microbe, for example, the Clostridium cluster and some short chain fatty acid producer. We then examined their metabolic pathways association of their major measures, and we found a total of 39 signal pathways were associated with at least one of the measures. I put some typical pathways in this figure. For example, the pathway of S-leucine biosynthesis was enriched by their physical activity, but decreased uh, by uh, by their adiposity measures. S-leucine is an important metabolite in their glucose and lipid metabolism. This is consistent with the energy expenditure of physical activity. And we also found an enrichment in the pyruvate fermentation by physical activity is an important pathway in short chain fatty acid production. We also examined the EC enzymes, and consistently with the pathway analysis, we found some EC enzymes, for example, the EC26142 increased by physical activity. It is an important enzyme in the pathway of S-leucine biosynthesis. We then examined the modulation of the gut microbiome on the association between physical activity and body weight change. And as a first step, we examined the modulation of PICO1 and PICO2 on the 6 associations. We found, except for CRP, the other 5 associations were modified by at least one of PICO1 or PICO2. Based on this finding, we further examined the modulation of the top 10 loading individual microbes on their association. And although for each association, several microbes showed modification, but we only find the species of Alistypis putridinis have a consistent modulation. For example, we found the individuals with a higher level of putridinis have a better decreasing trend in the body weight change from age 21 to stool collection, according to the quintile or long-term physical activity. When we summarize the association, we found those in the in the group of high putridinis and each 15 mass hour increment was associated with 2.4 kilograms weight loss or less body weight gain. But in the other group with a low putridinis, the number is 1.1, and we observe significant interaction here. But we should note this is a long-term average, a long-term physical activity across over 26 years. And we also examine the other associations, and except for CRP, we found a consistent modulation from putridinis on the other 4 associations. In accordance with the long-term physical activity, we found the recent physical activity and the 6 months weight change, their coefficient is about 0.5 in the group with a high putridinis. We want to know what pathways in putridinis drive this modulation. We then examine the other pathways contributed by putridinis. Among the 127 pathways, we found 11 have a significant modulation on the association between long-term physical activity and the long-term body weight change. Among the 11, 6 of them were of metabolism. For example, the pathway of gluconeogenesis, fatty acid beta oxidation. And for the 6 pathways of metabolism, we further examined each enzymes involved in the pathway by ranking the contribution from specifically from putridinis and as well as their modulation. For example, for the pathway of gluconeogenesis and fatty acid oxidation, we found they have at least one enzymes have their contribution from Putridinus ranking first among all contributing species, as well showed a significant modulation on the association between physical activity and the body weight change of long term. And when we put all the results together, we found among the 6 pathways, 4 pathways have at least one of such enzyme. The 4 pathways, including the gluconeogenesis, fatty acid oxidation, folate transformation, and steroid biosynthesis. And these findings support largely it's the 4 pathways driving their modulation in L-stipes putridinase. We further validate this finding in the independent cohort MBS because MBS have a relatively younger age compared to their discovery cohort MLVS. This makes us able to prospective examine the baseline physical activity and the baseline microbiome in relation to their body weight change in subsequent 4 years from the year of 2013 to 2017. Based on this prospective design, we find the individuals with a higher putridinase have a significantly stronger association between their physical activity and the body weight loss or less weight gain compared to those with a lower putridinase. And this finding was consistent when we examine the recent physical activity in relation to BMI stool collection and the long-term physical activity in relation to long-term body weight change from age 18 to the time of stool collection. This finding in the validation cohort support our finding of the modification effect of L-stipes putridinase we found in the discovery cohort. So summary of this study, we found physical activity have a small main effect on the microbiome, but the microbiome may modulate the physical activity's association with body weight. And we found individuals with a high abundance of the specific species putridinase will have a stronger body weight response to physical activity. And we found the pathways in putridinase of the gluconeogenesis and the fatty acid beta oxidation drive this modulation effect. And we found this modulation relatively consistent in men and women. This finding may help to explain the variability in the body weight response to physical activity, and more importantly, it may shed some light on a development of their personalized body weight strategies. Our study has several strengths and limitations. For example, the discovery cohort of MLVS were relatively old, so this limited the generalizability of our finding and also limit our ability for a prospective design. And also this is an observational study, so we cannot rule out the possibility of remaining confounding. I want to appreciate the mentorship from my mentor, Dr. Mia Song, and the secondary mentors of Curtis Hoover, Andrew Chen, and everyone helped me in this project, and every member in the Song Lab, in Hoover Lab, and all the participants and staff of the health professional follow-up study and nursing study, as well as support from Harvey Feinberg Fellowship. Thank you very much.